Hello guys, welcome back to my YouTube channel Excellence Hub. Today I am going to discuss the topic blotting technique. There are three types of blotting techniques: southern blotting, northern blotting and western blotting. We will see it. What are those? This is the general representation of blotting technique. Firstly, we take the sample and then isolate DNA, RNA or protein. We extract it according to our requirement. Then do the electrophoresis. After doing the electrophoresis, the bands formed on the gel being transferred to the membrane. Then the membrane that has the transferred bands are being labeled with probe. Labeled probe incubation step is there. Then target detection. The target band that we want is being detected. What is basically blotting technique is the transfer of macromolecules such as nucleic acids and proteins to solid phase membranous support is known as blotting technique. Fragments of DNA and RNA molecules separated by gel electrophoresis are transferred to a nylon or nitrocellulose membrane in a process termed as southern and northern blotting. Southern blotting was introduced by Edwin Southern in 1975 to detect specific sequences of DNA in DNA samples. Other blotting techniques termed as Northern blotting is done for RNA detection. Western blotting is done for protein detection. Eastern blotting is done for post-translational protein modification detection. Southwestern for a DNA protein interaction detection blotting techniques. These are some of the other blotting techniques. Now I'm going to discuss the three blotting techniques: southern blot, northern blot, and western blot. Firstly, the cell. Firstly, we take a cell that have DNA, RNA, and protein pool of DNA, RNA, and protein. If I'm performing southern blotting, then particularly I need DNA. By extracting DNA from the cell, firstly I will go for restriction digestion so that the large strand of DNA is being fragmentized into small fragments. Then perform gel electrophoresis. Run the bands on the agarose gel. Bands, fragments of DNA are being separated on the gel base on the basis of its molecular weight. Low molecular weight will Low molecular weight band will be found at the bottom side and high molecular weight band found at the top of the agarose gel. Then these bands are being transferred. These bands are then transferred to another membrane, transfer separated samples to membrane. Then probe membrane, single stranded complementary DNA or RNA to specific sequence, that is restriction fragment. Then detect labeled probe on membrane. Then the result is there, the labeled probe is being detected. Specific band that is being labeled with the probe is going to detect on the membrane. Sample contains specific DNA restriction fragment, can measure fragment size and amount. This is all about southern blotting technique. Then if I'm performing northern blotting, RNA is being isolated from the cell, single stranded RNA is being isolated. Then that single stranded RNA is allowed to run on the gel, that is gel electrophoresis step will take place. On the agarose gel, strands, different fragments of single stranded DNA are being separated in the form of bands. Then these are transferred separated samples to the membrane, single stranded complementary DNA or RNA to specific sequence. Then detection step, in which detection of labeled probe on membrane is done. Sample contains specific RNA transcripts, example mRNA. It can measure fragment size and amount or level of expression. Then third technique is Western blotting technique. In this, protein detection is there. From the cell, we will take protein, then denature the polypeptide, then do the gel electrophoresis. For the protein, we do use the polyacrylamide gel, that is SDS page is being run. Sodium dodecyl sulfate polyacrylamide gel electrophoresis technique is being performed for protein detection. Then these transfer separated samples to membrane. Primary antibody is being binded to the fragments to specify polypeptide. Then use of secondary antibody is there to detect or amplify primary antibody. Then the detection step, in which on the membrane sample contains specific polypeptide is being detected using the labeled probe, can measure polypeptide size and amount and its level of expression. Now, the difference between the three techniques. In southern blot, target for detection is DNA. In northern blot, target for detection is RNA. In western blot, target for detection is protein. Sample preparation. In southern blotting, DNA extraction, enzymatic digestion by restriction enzymes is being done. In northern blotting, RNA isolation, denaturation using formaldehyde is being done. In western blotting, protein extraction, protein denatured with SDS, that is sodium dodecyl sulfate. Separation. In southern blotting, separation is done using agrogel electrophoresis. In northern blotting, separation is done using agrogel electrophoresis. In western blotting, separation is done using SDS page. Type of membrane used. In southern blotting, nylon is used. In northern blotting, nylon is used. In western blotting, nitrocellulose or PVDF membrane is used. PVDF is polyvinyl difluoride membrane. Probe. In southern blotting, nucleic acid probe with single stranded sequence homologous to target DNA is used. In northern blotting, probe is used as RNA, DNA, or oligodeoxynucleotide. In western blot, probe used is primary antibody, that can be direct, or primary and secondary antibody, that is indirect, conjugated to fluorophore or reporter enzyme. Detection method. In southern blotting, detection is done through X-ray film and chemiluminescence. In northern blotting, again detection is done using X-ray film and chemiluminescence. In western blotting, detection is done through CCD camera or LED or infrared imaging. Now, this is a short trick to understand and memorize the blotting technique with its detection molecule. Snowdrop. SNOW. S stands for southern, N stands for northern, O is just a connecting word, W stands for western. Drop. For southern, detection is done of DNA. In northern, detection is done of RNA. In western, detection is done of protein. Now, this is western blotting. There are two detection methods of western blotting, either direct or indirect. Direct detection is being represented in figure 2, that is rely on a labeled primary antibody. Indirect detection is being represented in figure 3, that require a primary antibody directed against the target protein and a secondary antibody directed against the immunoglobin class or subclass of the primary antibody species. See in the diagram, first figure 2, direct detection relies on a labeled primary antibody. There is a protein to which specifically primary antibody binds. At the FAB region, enzyme is being binded. Substrate binds to the enzyme and produce the signal. This is the direct detection method of western blotting. Another one is indirect detection method of western blotting. Figure 3. Indirect detection relies on a primary antibody and a labeled secondary antibody. What happens? There is a protein to which primary antibodies bind. At the FAB region, secondary antibody bind. Then substrate binds with enzyme and produce the signal. So difference between both the technique is in direct detection method only one type of antibody, that is primary antibody, is required. In indirect detection method two types of antibodies are required, primary antibody and secondary antibody. Now the applications. Firstly, we will see the applications of Southern blotting. Southern blotting technique is used to detect DNA in given sample. DNA fingerprinting is an example of Southern blotting. Southern blotting is used for paternity testing, criminal identification, victim identification. Southern blotting is used to isolate and identify desired gene of interest. Southern blotting is used in restriction fragment length polymorphism, that is RFLP. Southern blotting is also used to identify mutation or gene rearrangement in the sequence of DNA. Southern blotting is also used in diagnosis of disease caused by genetic defects. Southern blotting also helps to identify infectious agents. Now the applications of northern blotting. The technique can be used for the identification and separation of RNA fragments collected from different biological sources. Northern blotting is used as a sensitive test for the detection of transcription of DNA fragments that are to be used as a probe in southern blotting. Northern blotting also allows the detection and quantification of specific mRNAs from different tissues and different living organisms. Northern blotting is also used as a tool for gene expression studies related to overexpression of cancer causing genes and gene expression during transplant rejects. Northern blotting has been used as a molecular tool for the diagnosis of diseases like Crohn's disease. Northern blotting also helped to used as a method for the detection of viral microRNAs that play important role in viral infection. Then last is applications of Western blotting technique. It helped to analyze recombinant protein expression, help to detect contaminant protein, help in determining alcohol abuse by measuring carbohydrate deficient transferrins level in the blood. So that's all about the blotting technique, the three types of blotting technique that we have studied: southern blotting for DNA detection, northern blotting for RNA detection, and western blotting for protein detection. What are the technique, what are the differences, and what are its application? All of these topics are covered. Thank you. Please subscribe the channel, hit the like button, and do share the video. Thank you.